DNA, or desoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, where it called nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA. The information in DNA is stored as a code made up for chemical bases: adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 19% of those bases are the same in all people. The order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in certain order to form words and sentences.
DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. A nucleotide are arranged in two long strands that form a spiral called a double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of the bases. This is critical when cells divide, because each new cell needs to have an exact copy of the DNA present in the old cell.
Genealogical DNA test. A genealogical DNA test looks at a person's genome at a specific location. Results give information about genealogy or personal ancestry. In general, these tests compare the result of an individual to others from the same lineage or to current and historic ethnic groups. The test results are not meant for medical use, where different types of genetic testing are needed. They do not determine specific genetic diseases or disorders. They are intended only to give genealogical information. Procedure. Taking a genealogical DNA test requires the submission of a DNA sample. This is usually a painless process. The most common way to collect a DNA sample is by cheek scraping, also known as a buccal swab. Other methods include spit cups, mouthwash, and chewing gum. After collection, the sample is mailed to a testing lab. Some laboratories, such as the Human Origins Genotyping Laboratory at the University of Arizona, offer to store DNA samples for ease of future testing. All United States laboratories will destroy the DNA sample upon request by the customer, guaranteeing that a sample is not available for further analysis.
Our conclusions. DNA and RNA are nucleic acids. Polymers found in all the living cells. They are formed by individual parts, divided in three classes: pentose, sugars, phosphate, and heterocyclic amines, bases. The phosphate group. A phosphate group forms the structural framework of the nucleic acid. It's a backbone composed of all alternating sugar and phosphate groups. It is attached to the fifth carbon of the sugar, and one nucleotide forms an ester bond.
Pentose sugars. Two types, ribose and deoxyribose. Difference: OH for ribose, H in deoxyribose. The only difference: the alpha and beta designations are interchangeable.
Heterocyclic amines. Sometimes called nitrogen bases or simply bases. Two major purines, adenine and guanine, and three major pyrimidines, cytosine, uracil, and thymine, are called bases because the amine groups have a basic property on water.
The complete structure. DNA contains thymine, but not uracil, while RNA contains uracil but not thymine. To remember the monomers of DNA, the amazing gene code TAGC.